Hi dear students, a warm good morning to all of you, and today we are going to talk about the staining techniques. We were discussing previously microscopy, and of course we know that when we view organisms, especially microorganisms under the microscope, their visibility as well as the clarity of the image becomes more better when you stain your specimens, because a glass slide is being found to be transparent, and the microorganisms also mostly they are being found to be microscopic, and they would be too tiny and microscopic in nature. And in such a thing, to view a microorganism uh, within a glass slide would be quite uh, difficult if you do not stain it. And in this session, we would be talking about the common staining techniques as well as an overall of what are the different staining techniques. So you can see that you can stain different types of organisms using different stains. When we view under the microscope, the organisms would be, if they are being found to be colored, we could easily distinguish them from the background, as well as the shape of the microorganisms could be identified with the help of staining them. Now, if you're going to look into the different types of staining techniques, you can see that if you do not stain a bacterial cell under the microscope, uh, you have to visualize the microorganism under the microscope with the help of your own eye, and you have to demarcate it from the environment, from the surroundings. So if a stain is not being put, sometimes you might see a situation as if it is not being, the bacteria or the microorganism is not being present. Even if it is present, you might not notice it. And if you do not have a stain, now this is a situation in which you have not put a stain, the environment could be like this, or the microscopic view could be like this. Now, staining process can be done either you're putting, no, this is a situation in which no stain is put. And sometimes you might use for microscopy a slide which has been stained with a single dye. So what happens? You can see all the organisms which can take up that stain will appear to be in that color. And the technique in which you use a single dye or a stain to stain the microorganism is referred to as simple staining. Then another staining technique which we have is you do not stain the microbe, but in turn you stain the background, or the glass slide is completely, or the background is being stained. And so what happens, the background will be appear colored, and against the background you have the microbe which is not being stained. Such a case is referred to as the negative stain. Other than that, we also do have a differential staining technique in which the same slide is being stained with two different stains. So, in such a case, some microbes will take up one stain, and the other microbes which have been present in the same slide could take up the different stain, a different stain. As a result, if you go to look into this slide or this microscopic view, you can see there are two different types of microbes which have been present here. And such a technique is called the differential staining technique. So you have a simple stain where you use a single type of dye, a negative stain where the dye is being used but it is not staining the microbe, but it is staining the background. And then you have a differential staining in which you are using two different stains, in which two different organisms will take up two different stains depending upon their uh, properties. So this is the overall of the different types of staining techniques which we have. But before going into the staining, we would have to do the preparation of our slide, and that's what we're going to talk about now. Now, if you're going to take a microorganism onto a microscopic slide or a microscope slide, what will happen? You take a lookful of it and you spread it on the glass slide. But if you just put that the organism or the loopful of the organism on the glass slide, and if you do not fix it onto the glass slide, what will happen? When you put the stain, as the stain is also a liquid, it might just flow off and go. So what you have to do is you need to fix it. Now, what is fixation? Fixation is a process in which the internal and the external structures of the specimens are preserved and fixed onto a particular position. Now, what is the position over here? The position which we consider here is the glass slide. And during the process of fixation, what would happen? You would inactivate the enzymes that might disrupt the cell morphology, and also you toughen the cell structure so that they do not change during the staining and observation. And normally, the microorganisms get killed and get attached to the microscopic slide during the fixation process. Now, how can you do this fixation? There are two different modes. You can either give a heat fixation or you can give a chemical fixation. As the term implies, the heat fixation is a technique in which heat is being applied so that the bacterial cells or the microorganisms gets fixed onto the glass slide. And in the case of bacteria and archaea, we routinely use heat fixation, and the overall cell morphology is being preserved, and you inactivate the enzymes over here. But you can see that, well, as you know, proteins are being heat sensitive. So the proteins get destroyed, as well as subcellular structures also might get distorted by the heat fixation. Now let's just have a look on how it is. You just take a liquid on the slide, that is a loopful of your culture or your broth, or your liquid or the source of your bacterial source. You take it and you prepare a loopful of that and you spread it over the slide at a particular area, and then you air dry it, heat gently. This is how you prepare a smear. Okay, so during the preparation of a smear also, you will have to heat fix it. Of course, you will have to keep the glass light intermittently on the flame, not always throughout for a lots of time, because the glass light might break. Just intermittently you have to just place it over the flame and immediately pull it back, and like that, so that the cells get heat fixed onto the particular slide. So for most of all the staining techniques, we do a heat fixation so that the microorganism is being killed and inactivated, and it gets adhered onto the particular glass slide. Now comes the method of chemical fixation. Chemical fixation is used to protect the subcellular structures as well as the morphology, and sometimes you can use chemicals like ethanol, acetic acid, mercury chloride, formaldehyde, as well as glutaraldehyde can be used. And these chemical fixatives, they penetrate into the cell and react with the cellular components, usually the proteins and the lipids, and they will be made inactive, insoluble, and immobile when they are being kept on the glass slide. So most frequently we use a heat fixation, but we can also use in a chemical fixation when heat fixation could distort the delicate structures which you want to view. Okay, or even the subcellular structures which would be damaged by the heat fixation can be viewed by the chemical fixation. And some cases you also have delicate organisms which would be lost by the process of heat fixation. So in such cases you can use some chemical fixation. Now let's just give a note on the dyes and the stains that which we use for a staining technique. Okay, if you go to consider the stains as well as the dyes, okay, they are synonymously used. They have some features in common. Usually the stain contains a chromophore group, which is a chemical moiety which with conjugated double bonds that will give the dye its color. and they bind the cells either with the ionic, covalent, or hydrophobic bonds. And you can see that, so two things are there in common. These dyes, okay, on the glass slide you have your fixed microbes, and now you're going to put your stain. And in this stain, there is a color giving component, okay, that is a chromophore group, and that can create conjugated bonds, double bonds, okay. And sometimes these stains can also have ionic, covalent, as well as hydrophobic bonds with a microorganism. And most commonly used dyes mainly are those which have ionic interactions with the microbes. Now, if you go to consider the ionizable dyes, they are of two types, either basic in nature or acidic. So you call them as basic dyes as well as the acidic dyes. Now, just as we think, be positive always. The basic dyes always have the positively charged groups. Okay, and they mainly bind to the negatively charged molecules such as nucleic acids, proteins, as well as the surfaces of the bacterial and the archaeal cells. The basic dyes are the ones which we most commonly use in the case of microorganisms, because the surface of the cell has been found to be negatively charged. Okay, and so if you want to stain a microorganism, what happens? You give a dye which has positively charged groups. So basic dyes are the ones which have positively charged groups in it, and they are mainly being used for the staining of microbes. And examples, you have methylene blue, basic fatsin, crystal violet, saffron, and malachite green, etc. And if you go to consider the acidic dyes, they are also in their ionized form, and they have a negative charge and they bind to the positively charged cell structures. Examples, we do have eosin, rose bengal, acid futsin, and all that. Okay, so let's move on. So as I mentioned earlier, the basic stains have positive charge and they bind to the negatively charged molecules of the bacteria, and the acidic stains have a negative charge and they bind to the positively charged molecules of the bacteria. And the cell surface is being considered to be as negatively charged, and as a result, what? Basic dyes are the most commonly used staining techniques. So I guess we will stop right now, and in the next session we will be continuing the types of staining, the different types of staining techniques like the differential staining and all that. Different staining techniques would be explained in the coming session. Thank you for now.